Hi, I'm Iris. I'm a master's student in the lab of Drs. Lorenzo Ferri and Vitin Sawan. My research is titled Organoids in Personalized Medicine, Alternative Treatment Options for Chemotherapy-Resistant Tumors in Gastro-Esophageal Adenocarcinoma, GEA. GEA is a type of cancer that appears at the lower end of the esophagus, the top of the stomach, or at the gastro-esophageal junction. The 5-year survival rate is less than 20%, making it the fourth most deadly cancer globally. Previous mutational analysis suggested that gastric and esophageal adenocarcinomas share the same genomic profiles and therefore should be considered a single disease. From 2007 to 2009, Dr. Ferri and team conducted a phase 2 clinical trials looking at the efficacy of docetaxel-based triplet chemotherapy for patients with locally advanced adenocarcinomas of the upper GI tract. They found the 3-year overall survival was improved to 60%, and this chemotherapy is a safe, tolerable, and highly efficacious treatment for GEA patients. Therefore, docetaxel-based triplet chemotherapy has been used as the standard of care treatment here at the Montreal General Hospital. However, around 40% of the patients have innately resistant tumors, and half of the initial responders develop acquired resistance during the course of chemotherapy. Currently, little is known about the molecular mechanisms of this process. In the last decade, patient-derived organoids showed great potential as a model that retained the histological pattern of the primary tumor and predict treatment response. But GEA tumors have not been investigated in such a model. Therefore, our first objective is to establish GEA patient-derived organoids as a reliable in vitro model that recapitulates patient response. We collected tumor tissue from GE patients at the time of biopsy and surgery. Then PDOs were established and treated with the chemotherapy agents. The ex vivo drug response was correlated with inpatient pathological response. The second objective is to identify targetable genomic and transcriptomic alterations driving chemoresistance. We perform whole exosome sequencing, bulk RNA sequencing, and single-cell RNA sequencing to identify candidate molecules. This was followed by high-throughput drug screening to validate the new targets. There are 30 patients in this cohort, 10 good responders and 20 poor responders. Patients had either complete or near complete pathological response were considered good responders. As you can see in the scan, good responders showed shrinkage in tumor after treatment, while poor responder had disease progression post-treatment. We also successfully generated a library of 29 organoids, 15 from treatment-naive endoscopy and 14 from chemotherapy-treated resections. Histologically, both organoids and primary tissue exhibited luminal structures with various degrees of filling. Intratumor heterogeneity observed in the primary tissue is also conserved in the organoids. Additionally, preservation of the clonal population in primary tumor and resulting organoids indicate genomic recapitulation of this model. Then we look at drug response. The heat map shows the cell viability of organoids after 72 hours of treatment. Good responder organoids have less cells viable after treatment, meaning they're sensitive to the chemotherapy drug, while poor responder organoids exhibited resistance with higher cell viability after treatment. And the differences in cell viability under these 3 critical concentrations were all significant between good and poor responders. This shows that our PDOs recapitulate patient response, representing a valid model for ex vivo drug testing. And after knowing the model is valid, we go on and look at the sequencing data. Here is a patient tumor showed amplification of EGFR, CDK6, and MET. And interestingly, in the following drug screens, we found that organoids are resistant to oxaliplatin, which is a chemotherapy agent, but sensitive to gefitinib, which is the inhibitor for the amplified EGFR. So for this patient, targeted therapy may be a better treatment option than traditional chemotherapy. And due to the heterogeneous nature of this disease, we also look at the expression profile at the single-cell level. The UMAP demonstrated a substantial shift in the expression profile of the epithelial cells in post versus pre-treatment samples. We found some cancer cell stemness markers are related to patient response. Patient A, who had upregulated stemness genes after treatment, experienced recurrence within 6 months after surgery. On the other hand, patient B showed no change or even decrease in this gene expression level, remains disease-free 18 months until today. So the next step is really to study how these markers really involved and contribute to the development of chemoresistance. Understanding the molecular basis of chemoresistance are of fundamental importance to establish predictive and prognostic biomarkers for GE patients. And our organoid provide a reliable model to investigate therapy resistant and predict treatment response in GE patients. This time, these organoids no longer need to be paranoid because they do function like little organs and help us design personalized strategies for chemoresistant patients, leading to a better overall patient outcome. Here is my reference and my acknowledgement. Thank you very much for listening.
I have a question, if you don't mind.
Yeah, go ahead.
If you can just speculate why this esophageal gastro-esophageal cancer, especially the adenocarcinoma, one of the squamous cell, it's so deadly. Christopher Hitchens, who was one of my heroes, died of it, and it happened very fast. The prognostic, it's really abysmal, and it puzzled me at the time, and it still puzzles me. Why is that?
I think one important reason is this cancer is really asymptomatic. So by the time of diagnosis, when people realize something, it's really at a very late stage. And secondly, because the esophageal adenocarcinoma is much more heterogeneous than any other cancer of the GI tract. So I think that's another reason that's contributing to the very bad overall survival for this disease.
Thank you very much. I think there should be more awareness on this type of cancer, because I think there is kind of really going under the radar a lot.
Yes, I absolutely agree with you. And a lot of researchers actually are looking at biomarkers that can help people identify this cancer more early on instead of at a very late stage.
Yes.
Iris, very nice work. So of the two patients that you showed, can you comment on their expression levels of HER2 gene?
Of the HER2 gene, okay. I picked my whole cohort. They are all HER2 negative.
Yeah. Okay.
Yeah, because clinically for this cancer, if you're HER2 positive, they would not only use the chemotherapy, they would also use the anti-HER2 antibody, which is the trastuzumab, as a therapy. But for my study, I only focused on those patients who received only docetaxel-based triplet therapy, chemotherapy. So they're all HER2 negative.
Oh, that's interesting.
Yeah.
Yeah, I think if there are no more further questions, is there a question in the chat?
No, we have the evaluation form for Iris in the chat, so
okay, great.
you can take time and fill it out for her. That would be great.
All right, if there are no further questions for Iris, Rahul, I think you are introducing our main speaker.
Definitely, I'll do that. Thanks a lot, Dr. Mawale. So it's a pleasure for me to be able to introduce Dr. Kapoor. I mean, people in the field, he doesn't need any introduction. He is a very great scientist and he does amazing work in the field of osteoarthritis. So Dr. Kapoor is the co-director of Schroeder Institute, which is the largest multidisciplinary arthritis institute in Canada. And he has had his training all over the world, including India, UK, New Zealand, Canada. And he has a Montreal connection, and he was a faculty member at UDM at one point of time before moving to University of Toronto. He's a world leader in arthritis research and is currently a Canada Research Chair Tier 1 and a professor of orthopedic surgery at University of Toronto. Dr. Kapoor's translational research program is directed towards understanding the complex cellular and molecular mechanism associated with joint destruction during OA and identifying reliable biomarkers for early identification of patients with OA and intervention. He also works on identifying novel therapeutic targets to stop and delay osteoarthritis progression. His research program is currently funded by CIHR, CFI, NSERC, the Krembil Foundation, the Arthritis Society, Stem Cell Networks, name a few. He also sits on the review panels and boards of various research and funding organizations across the globe. He has over 100 research publications in highly ranked scientific journals. So without further ado, I will just invite Dr. Kapoor to tell us more about his research and just shows the amazing work that he does. Dr. Kapoor.
Thank you, Rahul, and thank you for the opportunity to speak today. I'm really delighted. Of course, I'm not in Montreal today, but I'm present virtually. So good afternoon, everyone. I hope you're able to hear me properly. Today, I'm going to be talking about utility of high-throughput multi-omic strategies, and can we utilize these strategies to understand, or at least the attempt to understand, pathogenesis of a disease which is quite complex, such as osteoarthritis. These are a few of my disclosures. Now, if you look at osteoarthritis, of course, it's a very costly disease, especially in Canada. You're looking at billions of dollars which are spent on healthcare costs associated with osteoarthritis. It's a quite complex disease. It's now categorized as a serious disease. It's quite heterogeneous in nature. It's multifactorial. It's multidimensional, multi-source, and multi-origin. There are a lot of multis in this. But the real issue is in osteoarthritis, there are a lot of things about this disease we really don't know. Now, we know that if you look at last 2-3 decades, the lifespan of a human being has actually increased. While this is good news, we are all living longer, but it also comes with bad news that we are living longer with disability. If you look at diabetes or osteoarthritis, such diseases have really shot up in past 2 to 3 decades. And a number of years we're living with disability because our lifespan has increased, has considerably enhanced. And while it's great news, we are living longer, but it's also the major cause of disability. The major challenges we face in the field of osteoarthritis is that there are so many phenotypes in osteoarthritis. Some are clinical phenotypes, some are molecular and cellular phenotypes. How do you really marry and match these phenotypes is a real issue. Then we don't have any disease-modifying drugs which are approved at the moment which can stop the disease process. We only have symptomatic relief drugs, and that's why no DMODs actually exist which are approved at the moment. We also don't have any specific or reliable biomarkers which can help us detect this disease early on. So there's a lot of challenges and many more which I'll discuss in my talk. So the idea around understanding osteoarthritis, it's not a linear process because it's quite heterogeneous disease. There are so many different predisposing factors. You have age, BMI, ethnicity, female sex, genetics. And then on top of this, if you layer like joint injuries to metabolic diseases, bone deformities, and many more, all result in a really heterogeneous disease where we sometimes do not have an understanding of what the exact phenotype it is, whether it's the molecular, cellular, or the clinical phenotypes. And Francis, who's one of the leaders in our field, has really elegantly put this top-down phenotyping approach and step-up phenotyping approach to be able to really help us understand what these clusters of patients are, what their molecular or cellular phenotypes would be, and how we can start understanding this complex pathogenesis of osteoarthritis. Now, the complexity is not just at the patient level, because if you look at the number of tissues in a joint which are affected by osteoarthritis, let's start off by articular cartilage. In articular cartilage, this is one of the major tissues which degenerates over time in osteoarthritis. And in the articular cartilage, you have specific cells, chondrocytes, which respond to a wide variety of stimuli, whether it's inflammatory, catabolic, anabolic stimuli. And it really is a vicious cycle of these inflammatory, catabolic mediators, which really facilitate cartilage degenerative process. Now, if you look around the adjacent to the articular cartilage, you have another complex tissue called synovium. Now, within the synovium, you have cells such as fibroblasts like synoviocytes, or simply call them synovial fibroblasts, or you would have macrophages. These cells behave very differently to the way the chondrocytes would behave. Their cell survival, cell death mechanisms are very different. So now you have two tissues which are adjacent to each other, but actually have very different and complex cell survival, cell death mechanisms. And it makes it very tricky, especially from a therapeutic point of view. How do you target cartilage and how do you target the synovium? Now, on top of this, if you overlay metabolic complexities, people have different lifestyles, they eat different diet, so the metabolic status of an individual is different. And there are so many other factors as well. The degree of synovitis, which comprises of synovial inflammation, or the amount of extracellular matrix, which is actually deposited in the synovium over time because of persistent inflammatory insult, or wound healing response, which has just gone wrong. And as a result, you have an excessive amount of ECM deposition in the synovium, which contributes to joint stiffness as well as pain. Now, in addition to all these joint tissues and processes, you have fat pads, skeletal muscle, as well as our blood and nerve supply, all contributing to the complexity which we see in osteoarthritis. And that's why osteoarthritis is not the disease of just one tissue, but it's actually multiple tissues which are present in the joint. And then on top of that, you also have the changes which are happening systemically, which also have to be captured when you're trying to understand the pathology of this disease. Now, if you look at disease mechanisms at the tissue level, you now have to go deeper into a single cell level. And recent emergence of single cell analytics, advanced bioinformatics, these tools are now helping us understand that there are some major cells which are present in these tissues of interest, but then there are also subset of cells which are crucial in the early to advanced stages of the disease. And some of these cells are highly active or diminished during early stage of a disease. And when the disease is propagated, there is a distinct cell subsets which contribute to advanced stages of the disease. And that's why, not only understanding these phenotypes at the clinical level, and I talked to you about different tissues involved in the pathogenesis of osteoarthritis, but also distinct cell types, which are all contributing to this pathogenesis of this complex disease. Now, the real question is, and this is $1 million question, is that can we utilize high throughput multi-omics technologies and start understanding distinct phenotypes of osteoarthritis patients? And the answer is not that simple, and it's not a linear process, like I said to you before. And during this talk, I'll share with you some of the attempt we are making to understand these complex phenotypes. The idea here is that the patients who we want to study, who have osteoarthritis during distinct stages of the disease, we want to select certain biological analyses, certain high throughput omic technologies, whether it's the transcriptomics, genomics, metabolomics, or proteomics, to really understand those molecular cellular phenotypes. Now, the real issue comes is that you have a very complex clinical data which is obtained from patients. Now, how are you going to correlate that with the biological analyses which you have performed? And that's where we are attempting to use advanced computational and machine learning approaches, and with the hope that this would help us categorize these patients into distinct clusters or phenotypes. Now, patients evolve and change over time. And that's why any patient cohort which we are now creating, or the biobanks associated with these patients, we are trying to understand these patients over a longitudinal follow-up time points. And that's why longitudinal analysis is so critical, because these things change over time and you want to be able to see a response of a particular treatment, of a particular surgery. And that's why longitudinal analyses, when you are combining the clinical data, omics analyses, and applying computational and machine learning approaches, is so critical. So I will share with you stepwise process how we are attempting to understand these distinct phenotypes. At some places, we have gotten some success. Nothing is prime time at the moment. And to be honest, it's taken several years for us and my colleagues to be able to set up this infrastructure around patient cohorts and few of the platforms which I'll share with you. So please take everything with a pinch of salt, because this is an attempt to understand a very complex disease where we do have some early discoveries we hope to translate into a clinical setting. Now, the first thing which we have done is we've tried to create OA patient cohorts from early to advanced stages of the disease. One of the cohorts which I would like to share with you, and I'll share with you some of the results as well, is the knee-OA cohort. We have taken patients with different radiographic grades of knee osteoarthritis. So we have patients who are all symptomatic, but have grade 0-1 radiographic knee-OA, all the way to grade 4 radiographic knee-OA. We have also created a facet joint or spine OA cohort, where we have categorized this cohort using MRI grading. And then we've also obtained specimens in this cohort, and histologically, as well as gene expression profile and high throughput technologies we've used to categorize this cohort. All these cohorts are ongoing. And like I said, we have longitudinal cohorts. Using these biospecimens which we obtain, we've created an orthopedic biobank where we use very stringent criteria to collect, process, distribute, and of course, biobank. We want to make sure that the data or the analyses we perform on these specimens which are obtained from patients are high quality. And all of these are banked in a very stringent criteria, standard operating procedures. And currently we have over 150,000 orthopedic biospecimens, patients with various grades of osteoarthritis, such as knee, as well as we have patients, samples from spine osteoarthritis, but also foot, ankle, hand, shoulder, elbow are also part of this orthopedic biobank. Now, the next step is, when you have these very well-characterized specimens, what can you really do about it? Of course, there are several techniques, from genomics to transcriptomics, proteomics, metabolomics. But the real issue is that we have tried several techniques. I'll just give you an example. We've tried performing a very advanced scale multiplexing of our biospecimens, but we've really failed in understanding or identifying a clear signature in our biospecimens. And the idea is that maybe one of the technologies, a single technology, may not give us a clue or a signature to understand a complex phenotype of a patient. And maybe we would need to combine these various approaches, various omics platforms to be able to understand the complex phenotype of patients. So to do this, we started to first create the Next Generation Sequencing Center. The real motivation to create the sequencing center was to be able to do sequencing in a cost-effective manner. And I have a great team of colleagues, starting with our biobank manager to our sequencing specialist, to a bioinformatician, our biostatistician, as well as computational analyst, who have really helped set up this pipeline and works in a very efficient and cost-effective manner. And you can do high throughput sequencing, whether it's bulk, RNA-seq, as well as microRNA sequencing, which I will discuss in a bit. The second platform which we have created is the Metabolomics Center, and Dr. Vinod Chandran, who's a rheumatologist, and Dr. Nikita Luby, who's an expert in the metabolomics analyses, have helped set up this platform. And in this platform, you can perform targeted analyses and untargeted analyses. Now, using these platforms, the next step is let's envision that these platforms are giving you a very high throughput data, but how and when we should be applying this particular techniques in our patient samples. So one of the very ambitious projects which we are doing at the moment, and I don't have the large-scale data to present at the moment, is using a multi-omics approach in matched biospecimens from NeoA patients. Now, in this particular project, because it's a matched biospecimens, that means from each patient you have their synovial fluid, blood, urine, as well as joint tissues such as articular cartilage, synovium. Now, all these tissues are being subjected to various high throughput techniques: metabolomics, next generation sequencing, as well as cytokine multiplexing. The idea behind using these three techniques is that in a patient, if we have to understand their phenotype, the most important thing from osteoarthritis and its pathogenesis perspective is we really need to understand what is the inflammatory status of a patient, what is the metabolic status of the patient, and what is the gene regulatory or epigenetic mechanisms which are involved in creating those specific molecular or cellular phenotypes. And that's why we have chosen these metabolomics, next generation sequencing, and cytokine multiplexing. In a certain subset of patients, we are also performing single cell microRNA sequencing as well as RNA-seq, with the hope that when we combine this data, we are able to understand or identify at least one or few patient phenotypes. Of course, this data is coming from 500 patients, and I hope you can appreciate the fact that from these 500 patients, all of these biofluids and tissues are matched samples. At the moment, we have completed our blood cytokine measurements in all these patient samples. We've also completed blood metabolomics. We've halfway through our blood sequencing. We're also performing the synovial fluid sequencing as well. This would be followed by urine sequencing. We are hoping that in 2022, we would be doing the correlation and data coding to be able to match the molecular data with the clinical data. And if we are able to get some phenotypes, then of course the validation studies using external cohorts would be performed. But one thing is for sure, what would be the real outcome of this study is the creation of a data repository, that all this high throughput data would be publicly available as soon as it's completed, so that other investigators across the globe are able to actually utilize this particular resource, these data repositories, to be able to validate their studies. Now, while this was a multi-omic project, and I'll specifically talk to you about single technologies where we have seen some success, or I would say early success. And like I said, please take with a pinch of salt. These are early investigations where we're doing a lot of validation studies. One of the molecules which we are really interested in are called microRNAs. These are small non-coding RNAs. And we've seen that they have potential as OA biomarkers. And we've performed a series of studies where we have seen early signature of these specific microRNA signature, which could help us understand the early or advanced stages of the disease. And the reason why microRNAs are important is because they are quite stable entities. They are easily detectable, and they're easily quantifiable. If you would like to read more about the microRNAs and their biogenesis, we just had a recent article which has come out, which talks not only about microRNAs, but also other non-coding RNAs, such as long non-coding or SNO or circular RNAs. And if you wish, you can actually read this online. It's already has been published. Now, talking about microRNAs and its relationship with the early to advanced stages of the disease. So Dr. Amanda Ali, who was a former postdoctoral fellow, now she has her own lab in Detroit, she used this cohort where she took samples, so blood samples from patients with early and advanced stages of the disease. And we also had an external cohort in collaboration with Dr. Tom Appleton from Western Ontario, where we utilized their samples as well from their biobank. And what Amanda did was she actually took these blood samples, she applied them to microRNA sequencing. And now, to our surprise, we actually found a very, very distinct microRNA signature in early versus late stages of the disease. If you look at the PSC plot, you would be able to see that the early stage set of patients segregate very separately in a one cluster, and then they are quite distinct from late stage patients. And if this becomes even more clear, if you look at the heat map, the early OA patients have a very specific microRNA signature compared to the late stage microRNA signature. And when this data was probed further by Amanda, what she identified was that there are 7 specific microRNAs which are highly upregulated in the early stage compared to the late stages of the disease. Whether these microRNAs are involved in pathological process or just trying to maintain homeostatic functions, we don't know at the moment, and that's what we are probing at the moment. While this study was going on, the cohorts we used early and late stage, these were cross-sectional studies. So Amanda, along with our senior biostatistician, who's also professor at University of Toronto, they joined hands and started to look at the NIH cohort. So this is a longitudinal cohort where we have patient samples, blood samples from baseline all the way to 8-year follow-up. We also have the clinical data in these patients, and when we looked at some patients who are fast progressors. So, for example, a fast progressor is that patient who start with grade 0, grade 1, Kegler-Lawrence scoring to grade osteoarthritis at time 0 or baseline, and by 4th year they've already progressed to advanced stages of osteoarthritis. Now you have slow progressor who took almost 8 years to progress from grade 0 or 1 all the way to grade 3 or grade 4 radiographic knee osteoarthritis. And we also had a set of patients who were non-progressors. That means they had grade 0, grade 1 radiographic knee OA, but these patients did not progress in that 8-year period. Now, taking the blood samples from a fast, slow, and a non-progressor, we performed sequencing. And what Osvaldo and Amanda identified that there are certain distinct microRNAs which are different from a fast progressor versus slow progressor and a non-progressor. But what was really evident, and you can see on the right-hand side in the heat map, is that not all fast progressors are the same. Not all slow progressors or non-progressors are the same. That means there is so much heterogeneity within the same group of patients who are exhibiting an advanced or accelerated OA episode compared to slow progressors or a non-progressor. And that's why it's so important for us to start subclustering these patients based on their molecular profiles, their cellular profiles. In this context, like I said to you, that the patients which we are trying to categorize or dephenotype, it's very important that, yes, we are trying to understand what is happening in the blood, what is happening at the tissue level, but we also would have to go a bit deeper to be able to look at single cell level. Now, one of the limitations from our biobank is that when we biobank specimens, in this case, we have used synovium from patients with knee osteoarthritis. These bank samples do not yield very good results when they have been subjected to just single cell sequencing. And that's why, to avoid that, we actually started using single nucleus RNA sequencing, because we are able to avoid that digestion step, as well as because these cells, these tissues have been banked for a period of time, we are able to subject these specimens to single nucleus RNA sequencing. I'm so excited about this study because this is a pilot study so far, but I can tell you the type of data which we are seeing in this study is that when you take synovium from patients from early stage to the advanced stage of the disease and apply a single nucleus RNA sequencing in these samples, of course, you identify large set of cells. But when you start looking at the signature of these cells, you start to see that patients who have early stages of knee osteoarthritis and patients who have advanced stages of knee osteoarthritis are quite distinct in the types of cell subsets they are presenting. I just want to divert your attention to these fibroblast clusters, which I'm showing on the U maps. Now, if you look at the fibroblast in the graph above, these are all the patients who have early stages of radiographic knee OA. Now, if you look at the bottom part, these are the patients who have late stages of knee osteoarthritis. So now you can see the cluster of fibroblast all the way from early to late stage, it's expanding. There are also certain subset of cells which are present in the early, but are then diminishing in the late stages of knee osteoarthritis. Now, if you look at macrophages, it also exhibits a very similar phenomena. There are certain clusters which are highly present in the early, then diminished in late, and there are certain clusters which are less in the early stages, are highly expanded in the late stages. And really, to be honest, we don't know what these cell subsets, their function are. We are trying to understand a lot of things in this, and trying to, first of all, validate the clusters we have identified. Now, this is just an example of the macrophages. If you look at these macrophages, this UMAP is even better because you can understand different clusters in this one. So we have identified there are cluster 0, cluster 3, cluster 5, which expand from early to late stage. And then you have cluster 1 to 4 that shrinks from early to late OA. And similar scenarios also seen in fibroblasts as well, that you have certain cluster which are present in the early and then shrink, or vice versa. And understanding what these cell subsets are and how they contribute to the early to advanced stages of the disease is so critical. To do that, we are taking these cells and trying to conduct advanced flow cytometry. We have some deep sequencing going on as well. And the most important and probably the most exciting aspect of this study is taking an animal model from early to advanced stages of the disease and performing spatial transcriptomics. So we have taken joints which are naive, some joints which have been subjected to surgery, and they exhibit early to medial stage of the disease, and then all all the way to advanced stages of the disease. And we are trying to spatially understand the profile of these cell subsets, but also build their transcriptomic and proteomic profiles. So hopefully we are able to at least see how certain cells, which are emerging from early to advanced stages of the disease, how do they really contribute to the disease process. The last study, which I will discuss, is around another microRNA. It's called microRNA 181, and a potential therapeutic target. At the moment, we are pursuing this in a very aggressive manner. We discovered this particular microRNA in 2016, and these were all discovered in facet joint specimens as well as in knee osteoarthritis patients. And then after we discovered this particular molecule, we started to realize this molecule is quite detrimental for the articular cartilage, and we performed mouse studies, rat model studies, where we found that blocking this particular microRNA was yielding therapeutic benefit. It was able to reduce the degree of articular cartilage degeneration. And matter of fact, we've just started trials, which are safety trial in horses. So this microRNA 181, and just very simple schematic, basically in the lumbar spine, and I'm specifically focusing here on the facet cartilage. In the facet cartilage, in response to any insults, whenever the degeneration starts, we start to see high levels of this particular microRNA 181A5P. This microRNA 181 is not only present in the articular cartilage. As the process of degeneration increases, you also see this microRNA being dished into the synovial fluid. Now, the idea behind doing all this therapeutic studies against this microRNA was that we took cells from patients, and we found that these cells from patients who which were treated with microRNA 181 were resulting in very high inflammatory and catabolic activities. When you take this microRNA and inject this microRNA into the facet joint, it's basically eating up the cartilage. And we wanted to create a specific therapeutic, and we used a particular technology called antisense technology. And the idea was we wanted to stop inflammation and the destruction of the articular cartilage. And we used series of strategies, series of injection strategies to deliver the microRNA blocker into a facet joint or a knee joint, and with the hope that we are able to stop cartilage from degeneration. And I'll show you one of the data, and we've seen similar results in our mouse model as well. This is rat model. In this case, we've used a very simple model. It's a needle puncture model, which results in considerable degeneration of the facet cartilage. Now, when we treated these animals with the microRNA 181 inhibitor, you can see on the right-hand side, the articular cartilage is quite healthy compared to what happens when you're giving a control inhibitor. And we saw this massive cartilage protective effect in the knee OA model as well, not only the spine OA model. While these studies are showing promise that this microRNA 181 could be cartilage protective, we have a lot to learn in this. First is we need to know how safe this drug is. We really need to understand pharmacokinetics. What is the adequate dosage of this? And like I said to you, at the moment, we have our ongoing safety study, which is happening in horses. And we hope that we can get a favorable data, which is coming out from this, and hopefully which would allow us to move into the second stage in the equine trial, which would be an efficacy study. We are hoping that this particular microRNA 181 inhibitor would be applicable to both veterinary as well as the clinical medicine in humans as well. So we're really right now eagerly waiting for the results which are coming from this safety trial. So sometime in early January to March, we are expecting to get the full detailed safety analyses on this particular therapeutic. So what I've shared with you all today is, of course, osteoarthritis is a complex and a serious disease with so many different phenotypes. Our attempt is to utilize various multi-omic approaches. Are we going to succeed by using these multi-omic approaches to understand the complex phenotypes of osteoarthritis? Time will tell. And also, we have some encouraging data in terms of microRNAs, specifically around their potential to be therapeutic targets and their ability to stop particular cartilage degeneration. But like I said, this is not prime time at the moment. We have a lot to do, a lot to understand their safety profile before they even go into clinical trial setting. Thank you all for listening to me. Happy to take any questions.
Thanks a lot, Dr. Kapoor, for the amazing talk. It was very, very informative and very well structured, and it just covered all the topics. And it just is thought-provoking to see that how from a tissue it becomes an organ system when we deal with osteoarthritis. So any questions? There are. Richa, are you going to moderate for the questions, or Dr. Mwale? Or should I go ahead?
Rahul, you can moderate.
Okay. So there is one question from Joanna. She is asking that, are there any special considerations to take into account for dissociation of tissue for single cell and single nuclear profiling? What are your recommendations?
Absolutely. Well, there are various technologies where different methods you can use to dissociate tissues. We have realized that when you are dissociating with the same process for both the single cell or nucleus-seq is able to actually yield better results and better number of nuclei than the number of cells when you use the similar process to do the dissociation. So the digestion protocols, there are so many different digestion protocols you can do for tissues as well as the dissociation, but I would first compare the yield you are getting. But I just have an inking, and it would be dependent on a type of tissue you're using, and it would probably be different from one tissue to the other, but you may get better yield for the nuclei because they are able to withstand a little bit more harsher conditions than what you would be getting at single cell. But by no means single cell is an inferior technology. It is a far superior technology. It's just that the limitations we have is that we do not want to sequence one tissue at a time. And we want to eradicate batch effects, and we want to ensure that certain samples which we have banked a few years back are used at the same time when we are doing other samples which are being sequenced. And you know, there are a lot of limitations in the biobanking procedures where you have certain subset of patients or a certain set of patients who are going to be you're able to extract their samples, say 2 years ago. And now, after 2 years, you have identified another patient from which you can obtain a sample from. And from that perspective, I would say I would go into more the easiest path, which would probably be doing nucleus sequencing rather than actually doing single cell. But you can use similar protocols for this, but you know, it will all depend on the type of yield you're getting.
The next question, Dr. Mawali, you saying something?
Yes, so Mohit, that is a really excellent talk, really wonderful. I was at the edge of my seat. It covers really wonderful. So DChain he was in Chicago, and he was working with microRNAs, and his were like 204, 211 microRNAs, and he was showing that they may be involved in maintaining joint tissue homeostasis. And but you are showing different ones.
Absolutely. Faxon, it's a great question, because I wish I had actually shown a map of these microRNAs. There are certain microRNAs which are highly crucial to maintain the articular cartilage homeostasis. But there are certain microRNAs which are highly detrimental in nature. And microRNA 181, we haven't seen to perform any homeostatic functions. We've seen that it actually is influenced by inflammation or catabolic process, but also influences inflammation and catabolic process. So from microRNA 181 perspective, it is a specific microRNA which is detrimental in nature. Whether it has other homeostatic functions, we really don't know. And I think these things would become clear. We've done a lot of mechanistic studies, but I think it'll become clear as we're doing these safety studies as well. We've tried to look at their signaling. We have looked at their downstream targets. This microRNA 181 has a specific target called zinc finger. Zinc finger 440. We've also looked at targeting zinc finger 440 as well. The problem is, while this is a direct target of microRNA 181, it's not conserved in mouse, rats. It's only in humans and chimpanzees. So we've not been able to really test the therapeutic application of inhibiting this downstream target of the microRNA, which is zinc finger 440. Now, the real complexity in microRNA comes is that microRNAs, one microRNA can have both protective and detrimental effects because of its multiple downstream gene targets. And that's where the real issue comes. And that's why we are quite worried as well when we are talking about these microRNA therapeutics. And even in case of microRNA 181, we have not seen any homeostatic functions, but they could be.
Okay. So another question, one of the common features of these mouse models of OA is that they upregulate RANKS2. Have you seen in your studies, since you are doing a whole, you're looking at almost everything in human? And I asked that question because I think there are some microRNAs that were found to bind RANKS2. And in your case, do you see any of that?
Not specifically we have, but we have not actually even looked at it, facts. And so the way it works is that, of course, we've looked at microRNAs. We've looked at microRNA signature, and using bioinformatics tools, we are building their gene signaling profile and the gene targets. Now, there are certain things which do end up popping up, and definitely RUNX would be part of that. But in the list of priorities, so if you're looking at top 10 gene targets, we haven't really, or even protein-protein interactions, we haven't looked at RUNX2 specifically.
Okay, thanks. Thank you very much.
So the next question is from Ovidio. He's asking, as a microRNA carrier, what did your group notice that works better? Plasma, lipid-based carrier, or lipid-free proteins? Can you please care to speculate as why?
I think I'm only going to speculate on this because I actually don't have a real answer to this question. We are still trying to understand and identify. To be honest, I don't know if I can tell you that there is a true carrier of microRNAs, but I could speculate lipid, but that would just be a pure speculation.
We will take one question for Dr. Philip, and then we'll move on to other questions from the students. Dr. Philip.
Oh, hi. Mohit, excellent talk. Enjoyed it very much. Thank you.
Hi, Adi.
Yeah, hi. Yeah, I just wanted to ask you, you have very elegantly described the microRNAs. Where does this microRNA 181 come from, the cell type, and where is its target? Any of that kind of data?
So I didn't know I was going to be asked all the tough questions. So Annie, at the moment we've looked at bloods. We've blood from patients. We've looked at articular cartilage. We've looked at synovium. We've also looked at bone. When you look at the expression level as such, the articular cartilage from patients who actually have very early stages of the disease, the expression level of this microRNA is very low. The chondrocytes seem to be expressing this microRNA at basal levels, even in healthy conditions. This microRNA shots up big time in the articular cartilage when OA progresses. In the synovium, we have seen staining for microRNA 181 as well. And in the bone, there is staining in the subchondral bone. You see the expression of microRNA 181. Now, whether the microRNA 181 originates from the articular cartilage, and I'm specifically only talking about the joint tissues here, we really don't know where exactly this microRNA originates from and which specific cell types are actually contributing to this. But when it comes to OA, we have seen that osteoarthritis episode, and as OA is increasing, the expression in the articular cartilage is probably the max, which is even higher than what is observed in the synovium. So we believe that probably articular cartilage is probably the major source, but from the origin point of view, I cannot tell you that this is the origin. Now, other scenario is this microRNA is also present in the blood. So systemically, if there are changes systemically, there is also a way that this microRNA could be fed in the joints as well. So there's multiple scenarios. So exact source, no clue.
Okay, so I was also intrigued that you very, very good. I know. I thought so too. It might be multiple sources, but I'm just wondering whether I missed it. Did you show any single cell sequencing or do any single cell sequencing from articular cartilage itself, or just chondrocytes?
Yeah. So
I was just trying to see whether we can relate any of these microRNAs to a molecular phenotype.
Absolutely. We are doing it at the moment. So like I was showing in our 500 matched project, so we have taken 500 from 500 patients. We have samples: blood, urine, synovial fluid, as well as tissues, articular cartilage, synovium, fat pad. And all these are being subjected to various different technologies, so metabolomics, multiplexing, as well as sequencing. Because we can sequence blood very easily for microRNAs, so we are profiling microRNAs in all 500. But we cannot perform single cell in 500 patients, and we cannot perform nucleus sequencing in 500 patient samples. So what we've done is we've taken subset of patients. So the data which I showed you was just from the synovium, where we're doing single nucleus sequencing, but from the same matched patients, with the 10 patient samples which I showed to you, we are also now performing the articular cartilage sequencing as well. So that data is not available at the moment because we have not performed it so far, but that's what is in the cards.
Thank you very much.
The next question is from Rashad, and he's asking, what are your considerations to study the subclusters within the early, non, and late patient groups
to characterize the different cellular markers and cell types in osteoarthritis?
Right. So first of all, there are lots of subclusters. And how do you choose which clusters to study? It's a really problematic. Recently, I had an exceptional bioinformatician who's just joined our team, and her name is Shabana. And what she's doing is, and I've been trying to understand from a bioinformatician perspective, because to me, this is all rocket science when it comes to, you are performing single cell or single nucleus sequencing. And now you have major cell types. You have fibroblasts, macrophages. Now, all these cells are now have different subsets. Whereas, of course, different cell surface markers. But we have never known that some of these subset of cells, how and when they contribute to the disease process. Now, what we're doing is we are taking the entire clusters as a whole to understand what the entire fibroblast population is distinct from early to late. Now, the other specific way we are probing this is that we are subclustering. Say we have 4 clusters in early, 4 clusters in late for specific macrophages. We are comparing each cluster to each respective cluster from the stage of the disease. Then after that subcluster, we are trying to create a transcriptomic profile. The transcriptomic profile is being built in a manner where we are looking at what are the signaling pathways which are common in early and late stage. Can we identify those specific transcriptomic signatures which are highly present in the early or highly or diminished in the late stage? Or is there a fluctuation in the transcriptomic profile between early and late? There are a lot of clusters in even other cell types. Adipose cells are there. There are neurons as well, endothelial cells, which the data which I haven't shown today, because we're still probing those. But we're trying to take a holistic approach. But of course, in this case, you need advanced bioinformatics to really help you choose the clusters based on the differences in the transcriptomic profile you see in cluster 1 to cluster 2, cluster 3 in one phase. Then you compare those clusters to cluster 3, 4, 5 in the advanced stage of the disease. So there are multiple ways to analyze. We're trying all, and let's see what comes out.
So the next question is from Yasin, and the question is, what is the difference between osteoarthritis and arthritis? And can we consider osteoarthritis as an inflammatory disease?
So I'll answer the second part of the question. Yes, definitely you can consider osteoarthritis as an inflammatory disease. There are some preconceived notions or, historically, inflammation and osteoarthritis were all segregated, and everything was thought about inflammation related to rheumatoid arthritis and osteoarthritis as wear and tear disease. We're all going to get old and we are all going to get osteoarthritis. And which is not the case, because if you look at osteoarthritis, almost everything what happens in rheumatoid does happen in a patient with osteoarthritis. Is the extent to which something happens in OA or RA is different. Now, you can say in rheumatoid arthritis, the amount of synovial inflammation you see probably is larger compared to what you see in osteoarthritis. But my orthopedic colleagues who are doing joint replacements can say that at the advanced stage of the disease, they are unable to differentiate what is OA and what is an RA joint. So that really is a realistic scenario. Now, if you look at different catabolic processes, we have same processes which are originating, whether it's from the articular cartilage or synovium. in rheumatoid arthritis, the systemic contribution is very important. The immune involvement is very high, but that doesn't mean that these don't contribute to OA pathogenesis. I can tell you that if you are having osteoarthritis in one joint, now the data is emerging that the contralateral joints, take an example of a mouse model, you start to see some degeneration in the other joint as well. And now in patients, there is also an indication that it may not be one joint involvement. When you have one joint which is affected by osteoarthritis, you may have other joints also affected by osteoarthritis, and there is a local to systemic link. So yes, rheumatoid arthritis is different than osteoarthritis, but there are processes which are common, but of course to a different magnitude. And that is a big challenge for us, because we've always fall back upon diseases which are well characterized, and rheumatoid arthritis, and of course we have treatments, and we've still not been able to identify disease-modifying therapies for osteoarthritis because of those some preconceived notions we have about a simple or a process like inflammation, where we think inflammation may not be that important for osteoarthritis. In my opinion, it's very important, and it should be regarded as an inflammatory disease, even if it's low-grade inflammation.
There is another question from Ovidio, and he is asking, at the data correlation stage with clinical findings in the next years and further going into phenotypes, do you think that a potential main causality will be inferred? Of course, it's speculative at this point, but if you get a pattern, what would point to that?
Causality.
Causality? Okay.
Causality. Yeah, because correlation doesn't mean causality.
You know, the real intent about doing these multi-omic strategies is to be really parse out, or at least identify, can we identify subclusters of patients? Now, you know, we need very large scale cohorts to be able to do this. We would have to have a lot of validation cohorts and well-characterized specimens obtained from these cohorts to validate this data. Like I said, we'll be lucky if we'll be able to identify even one cohort. We really hope that we find a very specific pattern of patients who exhibit a certain clinical profile compared to other set of patients, and we see a very clear, whether it's a sequencing signature or the metabolomics signature or even inflammatory cytokine signature. The real challenge is going to be the integration of these technologies coming together, and how are we going to make ourselves understand that, oh, you can have 3 or 4 microRNAs, you can have 3 or 4 genes, you can have 3 or 4 metabolites, and then you can have 3 or 4 cytokines. That is a challenge where I don't have any answer, and hopefully next few years, that's why it's so crucial for us to understand any of these phenotypes. Hopefully, we identify a cluster of patients where all of these technologies do make sense. But whether we identify that pattern, we don't know. If we do, the next step, of course, is going to be validation in a larger patient cohort, which would be critical. But really, the real challenge is going to take this data and apply it to the real world. And so lots of hurdles.
So I assume that's why you are opening the source of everything to just create the momentum of other groups throughout the world to pick up, and basically then you will be able to aggregate everything to kind of really get to a conclusion. That, am I right?
That is absolutely right. And like I said during my presentation, was that one of the most important things which will come out of this, if we don't identify the clusters, this data is going to be open access. If you need high throughput sequencing data, you really don't have to repeat in distinct stages of OA disease. And or if you would like to validate your data, you would at least have all this data available, so that as we are trying to characterize, sequence, or subject samples to all these different technologies, you would at least have one source to at least correlate your data. So that's for sure. We need a lot of data. And these are very early days. And in osteoarthritis, we are playing catch up game. And such important and central repositories in osteoarthritis are crucial. So this is just a small attempt to be able to really at least create a data repository.
It's extremely laudable, and I think what you're doing should be followed by a lot of other groups, because I think that's the way that we can really move into really identify causality of all things. So anyway, I was blown completely away by what you did, and I understood maybe 10% of reality of what you're doing, but it's amazing, the approach. Thank you very much.
Are there any further questions? I will ask a question. Dr. Kapoor,
Yes.
what comes to my mind is that I was doing some canine work in a disease, mucopolysaccharidosis, where there's overexpression of GAG. So our thinking was that if there is a genetic mutation that overexpresses GAG, we should have a thicker cartilage and lesser OA. But this orphan disease gets OA and everything. Because anyways, when I was doing MRIs on these stuff in these animals, just to see that what are the modic changes compared to a normal one, one striking feature was fat pack degeneration. And synovial thickness was there, and we were blown. And I was always telling my PI at that time that this is mimicking an RA-like phenotype or a causality, but it's actually an OA because we don't have any evidence of any RA-like involvement. So it was always a thing between us that why is it presenting as an RA? Well, actually, it is an OA. And the striking feature in between was the fat packs. What are your thoughts on the involvement of fat pack, adipokines, and what comes first? Is it the biomechanical load that is altered that alters the cells in the fat pad, or is it the continuous irritation from the synovial membrane that leads these cells to secrete adipokines that accentuate the problem?
Absolutely. All ask me a lot of tough questions, and I have no answer for this. But I will share the concept here, because I think it's important. That's why we are trying to dephenotype patients. We all are different, and that's exactly the way the origin of a disease and the molecular mechanisms also work. Now, let's take an example of somebody who has existing conditions which could be related to their muscles. Now, muscle dysfunction related to changes in a lot of endogenous mechanisms. We know now that cell death pathways, even cell survival pathways, you can name quite a few, from cell death, apoptosis or autophagy. All of these are dysregulated in an individual who actually would have muscle dysfunction. Now, all of these could have impact in multiple joint tissues. It could affect the fat pad, it could affect the synovium, subchondral bone, and articular cartilage in a very different manner. Just imagine autophagy is suppressed in the articular cartilage. The articular cartilage, if it's suppressed in the articular cartilage, the cells, the chondrocytes don't survive. But in the adjacent synovium, autophagy is actually activated, and it actually results in overproliferation of these synoviocytes. Now, same process, which is happening in the articular cartilage, in the adjacent tissue, and doing opposite profile. And the way the process takes place is very different as well. So talking about the origin, I don't know if the origin is from a particular tissue, whether it's from the articular cartilage, from the synovium, bone, or fat pad. But I can tell you all these phenotyping exercise would hopefully help us understand that there are certain individuals where the primary tissue of interest could be fat pad changes, resulting in changes in the synovium, leading to the articular cartilage degeneration. On the other hand, changes in the muscle, changes in the alignment, the mechanical changes leading to the articular cartilage degeneration, production of inflammatory cytokines or catabolic mediators, then eventually affecting the synovium and other surrounding tissues is another kind of phenotype. So this is exactly the origin, and how it starts is very important for us to understand. Now, the real complexity here is post-traumatic osteoarthritis, injury-related osteoarthritis. And then you can actually look at somebody like age-related OA. The origin of age-related OA could be very distinct to somebody who had traumatic injury. ACL, these individuals are able to or are predisposed to getting osteoarthritis in accelerated fashion. And these are the animal models we use as well. Even in the animal models, even if we carefully study, we don't know if just the once you induce or surgically induce osteoarthritis, is it the articular cartilage where the disease gets originated, or synovial inflammation, or other processes which lead to joint destruction? So very tricky scenario. That's why deep phenotyping is critical.
Yeah,
And I think
definitely.
many individuals with very different origins.
So, Dr. Kapoor, also in your patient cohorts, that the biospecimens do you segregate them based on BMI or metabolic diseases, because that can also have an effect on the amount of fat they deposit, the function of immune cells that they're doing. So do you have any like retrospective data before the tissue was donated just to segregate and put that as a confounding factor?
Absolutely. And when I say we have comprehensive data, I can never claim that we have the best data, or we have good data, because what happens is that you have to collect a lot of data from patients, and we are learning over the years because we've made mistakes of asking 2 million questions from our patients. But eventually we realized that actually these specific questions don't make much sense. And or other way around, where we really missed out an opportunity to capture certain data, and now we are playing catch up game. So from each patient, at least we have basic information, their demographics, to their past history as well, comorbidities, type of medications, of course, trying to establish their metabolic status as well. So anybody who has diabetes, to obesity, to what their weight is, all of these things are all captured in the data bank. And from each patient, when we capture, we use a system called DATOs. And the DATOs where we do the clinical data capture. And then we, of course, have these molecular profiles, which we are creating, whether for microRNAs or single cell. The challenge is going to be how, a patient with a certain BMI range, and that particular molecular profile, how that interface would actually talk to each other. These are, things I think about. I don't know how to create those interfaces.
Yeah.
We are capturing this data, but of course, it's quite challenging. And we have a lot of patient-reported data, and that's where we are seeing quite a bit of discrepancy in terms of, that if somebody says that they're experiencing this much pain, and then your pain scales come into perspective, and sometimes your molecular profiles, the clinical profiles don't match those patient-reported data as well. So it's really complex.
Very complex, yeah. So we have another question from Dr. Grant. He asks that if you have or are you planning to determine if anti-micro-RNA 181 affects pain pathways in OA?
Absolutely. Great question, Michael. This is one thing where in our initial studies, we actually really failed to capture pain. I had this exceptional postdoctoral fellow, he's a rheumatologist, Dr. Akihiro Nakamura, who helped us create this cohort along with spine surgeon, Dr. Ramprasad. And one of the first questions we wanted to ask from this cohort was, and the specimens we obtained in this cohort was, can we identify a certain set of microRNAs which have any relationship with pain or pain mechanisms. So for microRNA 181, when we injected microRNA 181 in the facet or knee joint, we tried to capture, we did some pain analyses as well, pain measurements from Von Frey to other tests. But we were not able to capture the effects properly. And that's why we just couldn't get any good or reliable data related to pain from these studies. Now, if you fast forward this from the microRNA 181 to other microRNAs which we are testing, yes, we do have patients who are categorized based on different scales, so NRS. And based on these scales, we are looking at patients who have high pain to low pain, and how these patients respond to surgeries in a 1-year longitudinal follow-up time point, where at baseline you express certain set of microRNAs. Do they actually correlate to pain, or does it actually relate to anything if a patient responds or non-responders to surgeries? So we are trying to understand certain set of microRNAs in terms of their relationship with pain, but specifically microRNA 181, we haven't been able to look at our initial investigations. In the horse trial, which we are just started actually, so Dr. Thomas Koch, who is at University of Guelph, there are certain pain assessments associated with this. We are going to look at certain pain molecules in the synovial fluid of the horses which are going to be injected with this microRNA 181. So these are certain set of studies which we are going to do. And of course, in the efficacy trial, if microRNA 181 proves to be safe, we do plan to look at kinematics and whether animals are lethargic when you intra-articularly inject them with microRNA 181. So those are the things we have done around pain, but at the moment we have not done any pain-specific microRNA 181. The assessments which we have done hasn't yielded us anything where we can say with confidence that microRNA 181 can also reduce pain.
Wonderful. So
I think Dr. Kapoor, thank you for a wonderful talk. I think we've exhausted you with our questions.
It's a pleasure. I just didn't have answer to anything, but I'm.
That's good.
Yeah.
So with this, we would like to thank you a lot, Dr. Kapoor, for the wonderful talk and the wonderful discussion. And I'm sure that the trainees and the students have benefited a lot from the discussion, and they have a newer perspective as to what the disease is. It's not a modic, single tissue disease. It's multifactorial. It is way complex than we initially thought, or we undersold it for years. So thanks a lot for all the insights.
Thank you so much. I had a great time.
Thank you. Dr. Mawale, are there any announcements?
Well, that's it. Thank you very much. Really wonderful talk and really excellent direction. I think if there's a place where machine learning would be applied, it's here. Really wonderful.
Thank you.
Thanks.
See you
Thank
later.
you, everyone. Take care. Bye-bye.